University of Information Technology and Sciences, Dhaka. So, before starting my presentation, I would like to thank the managing committee of this conference for accepting our research work and giving the opportunity to present my work here. Let's see the title, Anti-Inflammatory Potential of Chloroform Fraction of Curcuma wallichii and its phytoconstituents, insight from in vitro, in vivo and in silico studies. We all know that inflammation is a complex biological process triggered by different physical, chemical or biological stimulus and mediated by the disruption of tissue homeostasis. A chronic inflammation is associated with lots of complications, the serious disease, and it also causes some complications like brain fog, the incoordination of brain due to neuroinflammation, and also the insulin resistance, which ultimately causes the diabetes, the mother of all disease, and blood clotting problems also. Here you can see the inflammatory pathways. In a sense, we can classify it in two types, arachidonic acid dependent and arachidonic acid independent. For arachidonic acid dependent, here cyclooxygenase pathway, lipoxygenase pathways are involved. And if we talk about the arachidonic acid independent pathway, inos, PPAR, nucleofactor kappa B are involved for the inflammation generation. Why we are talking about the plant-based medication? Few ago already one of my professors, Dr. Ekamul Islam sir, already had explained about it. So necessity of plant-based medication, because plant offers you the harmless and minimum side effects. It is easily absorbed and metabolized, easily affordable and easily accessible. And nowadays intensive research has been done on it, and increased scientific studies and clinical trials also allow us to use plant-based medication. You already see here the risk factors of NSAID, non-steroidal anti-inflammatory drugs. It is associated with the liver toxicity, renal toxicity, also cardiovascular complication and respiratory complication. So we mention here the traditional medicine and the modern science are not rival but partners, each hold half of the key to the holistic healing. Here you can see the information about Curcuma genus and inflammation. Curcuma is widely known all over the world and comprising of almost 120 species and many bioactive compounds isolated from Curcuma genus already in preclinical trial and also in clinical trial. I tried to mention some compounds here and how they work actually. This is the example for some bioactive compounds isolated from Curcuma wallichii and their pathway, how they work, and their dose. It is collected from different journals during the literature review. Now come to my plant, Curcuma wallichii, and why I select this. In our country, there are 12 species of Curcuma genus, and Acanthoran is the taxonomic classification depending on the flower and the position of flower. So after being identified in 2012, the Curcuma wallichii was scientifically unexplored. So we did intensive in vitro and in vivo test anti-cancer potential, and alhamdulillah we published it in Journal of Food Biochemistry. So we saw that the chloroform fraction of Curcuma wallichii was good at reducing the oxidant and also showed potent anti-inflammatory, anti-cancer activity. So as there is a relationship between the anti-cancer and anti-inflammatory activity, so that is why we encourage to conduct this research after getting those results and good at anti-cancer activity. So I already mentioned that there is a close relationship between the anti-cancer and anti-inflammatory agents because each of the pathway potentiate each other when the body is suffering from chronic inflammation. Then lots of inflammatory mediators produced in our body, which ultimately cause the cancer. And cancerous cells also potentiate the process of the inflammation. So both are closely related. So natural compounds like curcumin, resveratrol also show both anti-inflammatory and anti-cancer activity. So aim of this study was already mentioned here, in vitro, in vivo anti-inflammatory potential, chemical composition analysis by analytical tools, and the possible try to recognize the possible drug candidates by computational techniques. Here you can see the methods of a study. First of all, we collect the plants, then prepared it. After that, we conduct preliminary phytochemical screening, in vitro anti-inflammatory test, and then in vivo anti-inflammatory test, GCMS, and obviously HPLC analysis. And then we select the compound on the basis of their peak area, related percentage, and literature review. Then we conduct molecular docking using AutoDock-Wiener, and the drug likeness properties by SUST and PK-CSM, and then we analyze the result and conclude it. So here is the way we prepare the rhizome, and then phytochemical screening. We found the flavonoid, steroid, alkaloid, also phenols, a very good amount in this extract. And here you can see the result of anti-inflammatory test. For in vitro test, we perform heat-induced RBC stabilization assay and egg albumin denaturation assay. And then for in vivo test, we perform the acetic acid-induced breathing test. And we can see the fraction almost reduces it. The percent inhibition for RBC membrane stabilization is more than 53%. And for protein denaturation, it also more than 45%. And in breathing test also 59 and 69, which is quite good for a fraction. And for formalin-induced leaking test, it is about actually 2 dose, low dose and high dose we used, and it's about more than 51 and 59% for both dose in early phase, and for late phase it is 46 and 54%. So here is the GCMS data. We get more than 100 compounds, and based on the peak and the relative percentage and literature review, we select 53 compounds and then conducted the docking studies. Here you can see the result. Our standard diclofenac sodium showed the value 7.5, and few compounds here, those gave the result more than the standard. Telerone is there, and the Gederone is there. You can see the different phytoconstituents here. And this is the table which is presenting the GCMS information, the retention factor and their relative percentage. And here is the binding pose, 3D binding pose with the target COX-2 protein. And here we mention the molecular properties of selected compounds. No violation of the Lipinski rule, and they are quite good in terms of their molecular weight, solubility, and lipophilicity also. And also, if we see the molar surface area, the results are quite good. And bioavailability score also found more than 50%, so that is quite good. And ADMET study of selected compounds, here we talk about the absorption, distribution, metabolism, excretion and toxicity profile using the ADMET analysis. And to strengthen our results, we also conduct HPLC analysis, and we found 3 major phenolic compounds, camphorol, quercetin and tranexamic acid, which strengthen our results. So for discussion, I would say the phytochemical investigation revealed the presence of different class compounds, and HRBC, heat-induced RBC membrane stabilization, egg albumin denaturation assay. In vitro test, we got very good result. And then peripheral analgesic activity, acetic acid-induced breathing test, and in also formalin test, we get good result. And from the in silico model, we will conclude that Gederon, xanthine, and one agipino derivatives also demonstrate good binding affinities also and favorable pharmacokinetics profiles. So extensive in vivo, in vitro, and integrated computational study demonstrate that this fraction has very good anti-inflammatory activity. So we have a plan to do further research on, because GCMS analysis gave us the idea about the non-polar compounds. Sometimes some bipolar compounds also come out by GCMS results, but we submitted this research work in scientific reports, and they suggest us to do the LCMS analysis. So we have a plan to do that, and inshallah further we have a plan to do the in vivo model extensively, classical in vivo model also. So this is what about I want to say, and I think lastly I want to say the one sentence about this research work, that we identified 3 compounds which has a very good binding affinities with OX2 and a very good pharmacokinetic profile. So further research work will be done on that compounds to analyze their extensive inflammatory response. So thank you very much. This is all about my presentation.
Thank you very much for your nice presentation. Now, the floor is open for questions. Okay, I think the floor is a bit tired maybe, because we have, lagging behind our time. I think you have done the work. You have targeted one plant, and after that you have received the data, and then you enter into the bioinformatic analysis. But sometimes we have seen a lot of work, including this session in this morning. That sometimes the people are doing work the bioinformatics first, and then go into the target into the in vivo work or the in vitro work. So why you have taken this path?
Sir, probably the purpose of the study is to extract some phytoconstituents. That is the target, and we use plant-based medication before blindly, but nowadays, and there's a lot of example that lots of compounds are isolated, extracted from the plants. So first of all, we want to check that is these plants contain any activity. So we want to screen out that first, and then we try to identify some compounds. So among those selected compounds, we conduct the bioinformatics.
Yeah, the point is, I think you are validating your work with the bioinformatics here. So I think it was successful, but only one fraction from the chloroform. Did you work with other, solvents as well?
Yes. We first of all we fractionated with anhexane, ethyl acetate, also chloroform also. Aqueous fraction was there. But I mentioned before that only chloroform fraction showed a very good properties in terms of anti-cancer and also antioxidant. So that is why we select this fraction.
Okay, it was a really good work. I think that's all from my side, and thank you very much for your next presentation. And you really? Yeah. Sorry. Yeah, I think one question. Can you please, sir?
Well, it's not a question, it's a kind of question and comment as well. I have seen a number of titles in the abstract book which are investigating on the bioactive materials and so on, including yours. So my query or comment is that how many of you have targeted to patent your products that you have identified, or how many of these long list of products that you have been analyzing in this country for different universities went for production or commercialization? This is my comment. I mean, any of you can answer to that. Otherwise, I don't see that. Where is this research is going on? It's fundamental research to publish it. Somebody else is patenting. Somebody else is making the product and commercializing it for the benefit of India.
Sir, thank you for the suggestion, sir. Sir, before answering your question, I would like to acknowledge, because I can see one of my professors is here, Dr. Ikramul Islam, sir. Thank you, sir, for growing up us and making us available for this kind of platform. Sir, I appreciate whatever you have done for us. So, sir, I would say to answering the question that in a conference in Joshua University of Science and Technology, the same question I asked to one of our pioneer pharmacists, Dr. Abdul Rashid sir from Dhaka University. So to make the patent is quite difficult and more difficult for the scientists from these countries like Bangladesh and low and middle income countries like Bangladesh. So sometimes they try to collaborate with the first world country scientists, and sometimes he said that they betrayed with us and they made the patent by their name, so no contribution is there. So it's quite tough for the patent, I think for countries like us and the scientists like us. So, sir, same question I asked to the professor, and professor replied like this. Because lots of compounds already identified from different sources in Bangladesh.
But in that case, why are you doing this research? I mean, if you are doing the research, you are isolating this bioactive compound and then somebody else is taking and making money out of it, then why are you scratching your head and then spending all the money for it? What's the benefit for this country and for yourself as well? Only publication? Is that?
So, as a new scientist, I would say the authority should take the power and should, I think, they can play the roles. And then take the pathway for us.
Okay, thank you, Muhammad Sabir Hussain, for your presentation. And actually, the question from our the plenary speaker of the of yesterday, Professor Dr. Tarikur Rahman, he is here with us, and the question he asked is very important. Why actually we people are doing our researches here, and somebody is getting the benefit out of it in terms of money or the commercial perspective. The question is why the translation of the research is not being addressed in the commercial sector. Maybe that is the policy gap of the universities. In the Bangladeshi universities, most of the universities doesn't have the IP policy, and that's the actually gap that we need to fill. And if that policy is done by the universities, maybe they need some sort of the research and innovation centers with the capacity of the technology transfers. But that kind of the administrative things are not being practiced in Bangladesh, and that's the reason. In one side we have the universities, and the other side that the industries and the business entities, but there is no bridging here. There is very little, only a few bridgings are being made. I think that's enough from the individual perspective, not the institutional, and maybe we need to fill it up. And a lot of university vice chancellors are here, and I believe this kind of initiatives are required for the actual growth of the knowledge-based economy representation and the development of Bangladesh. We need to transform the researchers into the commercial sector. Thank you, sir. Thank you for your presentation, and that's all from me. And I'd like to thank the organizers for giving me the opportunity to present to chair the session, and thanks to all the presenters in the session. Thank you from me.
তারিকের প্রফেসর ডক্টর তারিকুর রহমানের কোশ্চেনের অ্যান্সারে আমি আরেকটা জিনিস অ্যাড করতে চাচ্ছি। আমরা মনে হয় পেপার পাবলিশের দিকে একটু বেশি নজর দেব, যে খুব হারি, মানে খুব দ্রুত একটা পেপার হবে, জার্নাল হবে, এই যে কনসেপ্টগুলো নিয়ে যে আমরা যে দ্রুত পাবলিকেশনের চেষ্টা করে থাকি, সেই পাবলিকেশন থেকে আমাদের ডাটাটা অলরেডি এক্সপ্লোর হয়ে যায় প্যাটেন্ট করার আগে, বা আমরা যে রিসার্চটা করি সেটার ফাইনাল টার্গেট অ্যাচিভ করার আগে ইন বিটুইনে আমরা যে সেগমেন্টাল স্টাডি নিয়ে যে পাবলিশগুলো জার্নাল আর্টিকেলগুলো পাবলিশ করি, সেখান থেকে ডাটাগুলো ফার্স্ট ওয়ার্ল্ডে যেখানে দ্রুত কাজ হয়, যেখানে ল্যাব ফ্যাসিলিটিস অনেক বেশি, এবং তারা ফিনিশিং এটা রিসার্চের ফিনিশিংটা তারা খুব সুন্দরভাবে দিয়ে থাকে, এবং ক্রেডিটটা সেই সব রিসার্চারদের কাছে যায়, এবং সেগুলো সাধারণত ফার্স্ট ওয়ার্ল্ড কান্ট্রির ওয়েল ইকুইপড ল্যাব এবং এক্সপার্টিস আছে, যে কারণে আমরা শুরু করে দিচ্ছি, আমরা আইডিয়া তাদেরকে দিচ্ছি, আমরা মিডিল পর্যন্ত এক্সপেরিমেন্টাল রেজাল্ট দিয়ে তাদেরকে পরবর্তী ইয়েটা তাদের হাতে আমরা তুলে দিচ্ছি, যে তোমরা এই যে প্লেট বানালাম, খাবার দ্বারা দিলাম, এখন তোমরা খেয়ে নাও, বিষয়টা এইরকম হয়ে যাচ্ছে। যে কারণে আমরা আমাদের দেশে ফিনিশিংটা, এটা আমরা যে আমাদের তৈরি করা জিনিস, সেটা আমরাই যে আমাদের ক্রেডিট, বাংলাদেশেরই যে ক্রেডিট, বাংলাদেশের ইউনিভার্সিটিরই যে ক্রেডিট, স্টুডেন্টদের যে ক্রেডিট, এই জায়গাটা আমরা আনতে পারছি না। তবে হ্যাঁ, সময় আস্তে আস্তে অনেক দূরই এগিয়েছে, এখন আমরা আইডিয়া জেনারেট করতে পারি, অনেক দূর পর্যন্ত যাইতে পারি, কিছু কয়েক বছর আগে তো তাও যাইতে পারতাম না। আশা করি প্যাটেন্টের দিকে বা সেই ক্রেডিটের দিকে আমরা অতি দ্রুত আসতে পারবো, এই আশাবাদ ব্যক্ত করে আজকে সেশনে এখানে সমাপ্তি ঘোষণা করছি। ধন্যবাদ সঞ্চালককে, ধন্যবাদ অডিয়েন্সকে। আজকের প্যারালাল সেশন 4 এখানে শেষ। ওকে, ধন্যবাদ।
Speakers for their insightful and thought-provoking presentations, as well as the session chairs for conducting the sessions. We are delighted to have our chief guest of ICLS 2025, Professor Dr. A.M. Sararuddin Choudhury Sir, Honorable Vice Chancellor, Shahjahan University of Science and Technology, among us. Therefore, I would like to request to kindly come up on the stage and present crest of appreciation to our honorable session chairs, Professor Dr. Mohammad Shahidul Rahman Sir, Department of Biotechnology and Genetic Engineering, Jahangirnagar University, and Professor Dr. Shamim Ahmed Sir, in recognition of their kind presence and contribution in chairing this scientific session.